please everybody make some noises if it doesn't, if something goes horribly wrong.
Oh, and just whilst Chris is setting up, if anybody has any questions, if you could put them into the chat, then I'll keep an eye on them so that I can ask Chris at the end of his talk. Thank you.
Okay, thanks. So we'll make a start. So I'm going to talk to you a little bit about basically a protein that we've been working on for virtually the whole time we've been here in Bangor, and this is a protein that I characterized as a postdoc. First of all, it was a factor that at that point was an uncharacterized human open reading frame that was, we knew that it existed but had no idea what it looked like or what it did. So we've been trying to sort of figure that out over the past 6 years or so here in Bangor. And so I guess to go back to basics first of all, for folks who maybe don't have too much of a background in DNA repair, I'm sure most folks are familiar with the concepts of the radiation, as well as various chemotherapies. It can induce DNA damage, and this is really how most of these therapies function, by inducing either DNA breaks or modifications to the DNA backbone of cancer cells. And ultimately, once this damaging load gets to a certain level, any cell will die, and also in some cases very rapidly. Rapidly dividing cancer cells are particularly sensitive. So this is really where our research comes in, because there's a worldwide community really that are devoted to trying to figure out what the mechanisms of repair of all these DNA breaks and DNA obstacles, and the mechanisms by which those proceed. And this is really coming from the concept that if we can exacerbate the load of these of DNA damaging cancer cells, then we can really improve cancer cell kill to some degree. And so one of the sort of landmark discoveries that decades ago now in the elucidation of how all these DNA breaks are repaired came from the discovery that it can really proceed by either one of two main mechanisms. And this is a bit of a simplification, to be honest, for the purposes of this talk, but I think it suffices for what we need today. And so, some really seminal discoveries from the past have elucidated the DNA really repair really proceeds by two very specific and distinct mechanisms. So the most basic form of repair is known as non-homologous end joining, and this is an error-prone pathway that can lead to genetic changes that may indeed actually be pro-tumorigenic in some circumstances, and could be pro-cell death as well. And this is almost a bit of an emergency mechanism. These are kind of emergency mechanisms in a way, because if you've got a DNA double-strand break, it's very easy for those two ends to lose each other. And once they've lost each other, the chances of them finding each other again and going back together in the right order, it's actually pretty slim, and that's catastrophic really for the cell if you consider just how important its genomic sequence and structure is to its basic function. So if you have got a break in both strands of the DNA backbone like this, one of the first things that will happen is that there are subsets of proteins that will be recruited extremely rapidly to those broken DNA ends, and they will try and hold them together while the actual DNA repair can take place. And so some of the first factors are seen here: the cu-DNA PK holoenzyme complex in this end joining pathway, and these are really the first factors to go there almost instantaneously following a DNA break. But then subsequent steps in this end joining pathway simply involve the ligation of these DNA ends. And conversely, in the case of the second pathway, which is called homologous recombination, this is an error-free pathway which proceeds by a completely different mechanism. So we can see these mechanisms expanded here. We can see the several steps in religation of this DNA break by the end joining pathway on the left-hand side. So really these ends are being glued back together, but this can involve some processing and therefore loss of sequence. But on the right-hand side, we have this homologous recombination pathway, and we can see that this proceeds via a really distinct mechanism, which actually involves initial chewing back of some of the DNA ends here. So we can see one of the first proteins to go is this complex called the MRN complex, and this is really the sort of core focus of what I want to talk to you today about is this complex and how it is controlled. So one of the first steps in this homologous recombination pathway is mediated by this MRN complex, which actually contains the nuclease MRE11. And so nuclease is obviously an enzyme that digests DNA, so the first step that occurs is the digestion by MRE11 and other nucleases of one of the DNA ends beside the break, on either side of the break, to reveal a region of single-stranded DNA. And we call this process DNA end resection. And it's actually this single-stranded DNA that gets coded first by RPA and then by Rad51, which acts as a recombinase to actually invade the DNA strand of a neighboring sister chromatid. And this sister chromatid has got the correct DNA sequence to then copy back and make sure that the broken DNA sequence is fully repaired correctly. And if you think about it, this is a mechanism that must therefore be limited to stages in the cell cycle where the DNA has been replicated or is being replicated. So we have these very different steps. There's a simple sort of gluing mechanism on the left-hand side, which is error prone and can introduce can actually be mutagenic. And this error-free mechanism, which is much preferred by the cell, which uses a second healthy copy of the same piece of DNA to copy back the correct sequence and maintain the structural integrity and the sequence integrity of the genome. And how this really feeds into cancer biology and the biology of other diseases, but particularly how we treat cancer, is really based around sort of several different strategies. So, we know that most radiotherapies and chemotherapies are either working by inducing DNA breaks, or they're making modifications to the DNA backbone that interfere with DNA replication progression, for example. So what people have tried to do over the years is either to drug target certain DNA repair enzymes, which in some cases has been quite an effective strategy. So things like kinase inhibitors of master regulators of DNA repair, when those drugs are combined with DNA damaging therapies, or even sometimes as monotherapies, they can be quite effective potentiators of cancer cell kill. And likewise, there's a whole arm of this of work that's really associated with the development of precision medicine strategies to try and target tumors with particular genetic backgrounds and genetic defects, so that the treatment is essentially targeted to that genetic background to enhance the patient response. And a classic example of this are, as we'll see in a second, are the use of PARP inhibitors, so to treat BRCA deficient cancers. Okay, so this is not just a good example and sort of classic example of how you target a treatment to a specific tumor with a defect in an important DNA repair factor. It's also quite relevant for some of the work that I'll show later whenever we start to use some of these drugs. So PARP essentially stands for poly-ADP ribose polymerase. So it essentially builds these poly-ADP ribose chains at sites of DNA breaks and also at stalled replication forks. So these are essentially nucleic acid mimicking chains, which are very adept at recruiting lots of other repair factors to the area of the DNA break to elicit repair, or they are very important in the restart of stalled replication forks as well. But also PARP is part of a mechanism that repairs single-stranded breaks, and we talked about double-stranded breaks already, but single-stranded breaks occur extremely frequently in the lifetime of every cell on a daily basis, and this is really leads into one of the theories of how these PARP enzyme and PARP inhibitors function to selectively kill cells that are deficient in tumor suppressors like BRCA1 and BRCA2. So the theory is that cells with functional BRCA1 and BRCA2 deal extremely well with single strand breaks. Even when they're converted to double strand breaks, there's active BRCA1 and 2 in the cell to help promote the repair of these lesions. Conversely, if you find a tumor that with loss of heterozygosity of BRCA1 or 2, so with loss of function of BRCA1 or 2, and combine that with PARP inhibition, essentially there are these frequently formed double strand breaks are not dealt with well by the cell, and because of the lack of homologous recombination mediated repair associated with BRCA1 and 2 function, and these cells therefore preferentially die. So this is why led to the licensing of PARP inhibitors for a number of cancers now with impaired BRCA status. And this is sort of relevant to what I want to talk to you about today, because this is a nice valid mechanism that obviously is at play, but there's some recent research that's kind of called into question whether this is the only mechanism at play in BRCA deficient cells. So, as I mentioned a few slides ago, all the work I'm going to talk about today is based around the control of this nuclease, Emery11, which performs these resection events at DNA breaks. And just to elaborate a little bit further, so we have this very simplified version of the earlier pathways that we looked at, where we have a broken piece of DNA. So we can see where this Emery11 nucleus functions. So it can function in two different sites at DNA breaks to initiate this initial resection step. It can function firstly as an endonuclease to make a nick on either side of the DNA break, but then also as an exonuclease from the end of the strand to chew in from the edge of the break to create the resected ends that ultimately elicit DNA repair. And so this is where our protein of interest comes into play. This is the protein mernib, which we have worked over the past 6 years or so to try and elucidate its function. And most of the functions that we have elucidated are Emery11 associated functions. And so what we really think is, and the story I want to tell you today is just a little bit about how we think this protein controls Emery11. And mernib itself seems to be a very simple protein. It has at least superficially, it has an N-terminal zinc finger-like motif, which is predicted to coordinate a metal ion. And then the rest of it is somewhat unstructured, although there are several sort of sequence features that I'll delve into as we go across the rest of the talk. But what we've really been interested in is answering this question of how this nucleus Emery11 is controlled. So if you think about it, your nucleases are actually really quite dangerous things. This is an enzyme that cuts DNA and is functioning in DNA repair. Although if an enzyme like this isn't properly controlled, then it's pretty easy for it to sort of wreak catastrophic havoc on the human genome. And this goes for the multitude of nucleases that are present in the human genome. And for that reason, there are a lot of very specific and sort of complex and nuanced ways via which the activity of these nucleases is directed and limited. So I think this is where the study of other proteins and other factors like mernib, which function to regulate Emery11, becomes really important, because it provides a lot of information about how these quite dangerous enzymes can be directed properly and their activity can be reined in. Okay, so just to go back to the very early days of working on this protein. So we originally, even this was 10 years ago now, there was conducted a screen using siRNA to study to try and identify various genes in cancer cells that were associated with elevated levels of DNA damage. And this is where this MIRNIT factor was identified. So several of these screens were carried out by different labs around the world, and this one kept including work that done by my postdoc supervisor, Spencer Collis in Sheffield, and several other labs like Caroline Simpridge's lab in the States had conducted similar screens, and this MIRNIT gene was popping up in several of them. Following the identification of MIRNIT in the screen, we essentially conducted some mass spectrometry interaction analysis to try and really figure out what this protein is binding to. And the main hits were obviously the MRN complex. This is really what got us into the idea that this could be a novel regulator of MRE11. So we can see just some nice, this is a nice stable cell line expressing flag tag MIRNIT, and we can see it very nicely pulls down all three members of the MRN complex here. And more recently, we've actually gone on to make recombinant protein. For both MIRNIT, we've made recombinant MIRNIT protein in bacteria and recombinant MRE11, which is a lot more difficult in insect cells. So the only place you can really make full-length human MRE11 without it falling to bits or being insoluble is in the baculovirus insect cell system. So this is what we've done, and with the initial intent of trying to figure out to which part of this MRN complex does MIRNIT bind. Because of course, whenever we do the previous experiments that we've just shown, we don't really have any idea of which complex member it's binding. And so just to take this apart, we've conducted experiments where we essentially are binding these proteins together in vitro in tubes. And it is, at least in vitro, able to bind to MRE11 without RAD50 or MBS1 present. So we think it directly interacts with MRE11. And this kind of fits with what the other phenotypes that we see. And usually amongst DNA repair factors, we have the recruited to sites of DNA damage. This is one of the sort of common features that core DNA repair factors will very quickly get recruited to sites of DNA damage. And so to prove this is the case with MIRNIT, we've introduced these laser stripes using a laser microirradiator. So this is essentially a stripe of DNA damage across a live cell with fluorescently tagged MIRNIT here in the nucleus. And you can see even within 30 seconds of DNA damage being induced, we have a really nice stripe of MIRNIT localizing to the site of damage. And when we look a little bit more at the structural features of MIRNIT, we find that really one of the most interesting things that we spotted immediately was that it had a nuclear localization sequence, which is maintaining its activity in the nucleus where DNA repair needs to occur. And so this is mediated by this little run of lysine and arginine residues. And we find whenever we mutate these to alanines, so this sequence can't function anymore, we find this makes MIRNIT essentially flip out to the cytoplasm. And so you can see that here nicely again. There's a nice picture of this mutant version of MIRNIT with the nuclear localization sequence disrupted. And what's interesting about it is that if you actually co-localize this with members of the MRN complex, we've done it here with RAD50, you can see that the binding is actually sort of sufficient between the two to pull out RAD50 to the cytoplasm with it. And our initial work, which I'm just going to run through reasonably quickly before we move on to the more recent work we've done here in Bangor, essentially validated that in the context of ionizing radiation and just DNA double-strand breaks, what we're seeing in the absence of MIRNIT is actually less recruitment of the MRN complex to sites of damage, and also less activation of downstream pathways. So one of the main functions of the MRN complex at sites of damage is not just to conduct this resection event, which we're seeing here, but it also is required to activate the master regulator of the DNA damage response, ATM. So we also figured, well, maybe this is a function that MIRNIP also contributes to. And indeed, you can see this is the case, because if we look on the right-hand side here, whenever we ionizing radiation treat these cells, we see a really nice stimulation of ATM activity and phosphorylation of its downstream target CHEK2. But in the absence of MIRNIP, or in MIRNIP depleted cells, you can see this is really drastically impaired. So we're thinking there that the MIRNIP is essentially making all this happen essentially by helping MRN get to the sites that it needs to. And indeed, if we conduct a fancy assay based on double-strand break resection via qPCR, we can see that, again, there's a defect in resection itself. And this is all just in MIRNIP depleted cells, and that's consistent just with the previous observations that in the context of radiation, MRN doesn't always get where it needs to go in the absence of MIRNIP. And indeed, if we look a bit closer, actually cell survival. So these are I think HCT116 colorectal cancer cells. So we've depleted these cells of MIRNIP using 2 different siRNAs, and then treat them with an increasing dose of ionizing radiation. And you can see that both MIRNIP depleted samples are rather sensitive to the radiation. So it all fits with the nice picture that MIRNIP is actually really important for promoting DNA repair, and is doing so via its effect on the MRN complex. However, we also during all this time spotted that the MIRNIP deficient cells were also sensitive to hydroxyurea, and this is an agent that doesn't necessarily cause DNA double-strand breaks. So it's actually an agent that stalls the progress of DNA replication. So we really start to figure, well, is MIRNIP also functioning during DNA replication and not simply at sites of DNA repair? And this coincidentally sort of fed into something that was coming out in the literature at the time, was that in addition to the function of BRCA1 and BRCA2 in DNA double-strand break repair, there are additional roles for these proteins at sites of DNA replication that have been stalled for one reason or another. So basically, cancer cells, like any cell, have got to replicate their DNA before they can divide. And so to do that, they have to open up this strand and get the sequence information inside. And so that for that reason, you can see that whenever the process of DNA replication occurs, the 2 strands get opened and this replication fork-like structure results. So you can see this is the process of DNA replication in progress. And there are multiple things that can essentially get in the way of this process. And this is including but not limited to formaldehyde in cigarette smoke, acetaldehyde from alcohol metabolism, various pollutive chemicals, but also, as we mentioned before, lots of forms of chemotherapy which can interfere with the process of DNA replication. For a nice example would be platinating agents like cisplatin, which form intrastrand crosslinks between the DNA strands, and which therefore impedes the progress of the replication fork. So and these treatments essentially affect very rapidly dividing tissues in particular, as well as rapidly dividing cancer cells. So one of the emerging stories in the literature over recent years was the fact that one of the main responses to a block to DNA replication is actually for the cell to remodel its replication fork to essentially avoid that block. And you can probably think of it a little bit like avoiding a car crash. If you can see you're about to drive into a brick wall, you're going to hit the brakes. And so this is essentially what replication forks do when they sense an obstacle ahead. They actually remodel themselves, and we call this fork reversal, into a completely different structure. And you can see what actually happens as a consequence of this is that this kind of chicken foot-like structure is formed, but in the process, a new DNA end is created. And just like a double strand breaks, this DNA end can be acted on by DNA repair enzymes. And indeed, this is actually what proves to be the case. So again, we have this remodeled DNA end ahead of a replication blockage, and in the presence of BRCA1 and BRCA2 and all these fantastic tumor suppressors, there's no issue here because these factors actually will associate with this reversed end and protect it from the action of all the different nucleases in the cell which are out there. However, in the absence of any of these factors, BRCA1, BRCA2, or a number of other DNA repair factors, this end becomes deprotected and actually gets chewed up by the enzyme MRE11. And this leads to what we call fork degradation and eventual genome instability. So since it's MRE11 that's doing the DNA chewing in these various usually cancer types that have lost these essential tumor suppressors, we obviously start to wonder, does Mernib have a similar dual role in repair and replication? And so we start to turn to CRISPR as well, because all our work had previously been done with siRNA. So we made CRISPR-Cas9 made deletions of Mernib in 2 different cellular backgrounds. So this is HCT116 and HeLa cells, so colorectal and cervical cancer cells. And we saw very quickly just that both Mernib knockout clones that we had lots of extra DNA damage, and we could rescue this by reintroducing Mernib into the system. So it's nice because it shows it's not an off-target effect of CRISPR randomly chopping the genome elsewhere. And we also went on to do some really nice basic assays to characterize just how much DNA damage was in these cells under resting conditions. And we used a technique called COMET assay, where essentially the pieces of DNA in an agarose gel will travel further if they're broken. And so we measured the kind of tail of the comet, which and the longer the tail, the more DNA breaks there are. And you can see that by this assay that we do see more DNA damage. But also if we start to look at chromosome structure, so this is a nice normal happy chromosome and mitotic chromosome, and then here we have some abnormal ones where they've been fused together in various different ways. We can see that the frequency of both of those goes up in Mernib knockout cells. But then we start to really ask the question: what does Mernib do in conjunction with DNA replication? And so firstly, we use the technique called IPOND, which is essentially a way to figure out if a protein is associating with newly formed DNA. I'm not going to go into the method at all, but you'll have to take my word for it. But you can see in both of these assays there's quite a lot of association with Mernib and newly formed DNA. And you can see here, as a control, this is a nice sample where there's a lot of purified pCNA, and this is one of the factors that basically performs DNA replication. We then moved on to DNA fiber assays. So this is a novel technique that allows you to basically assess the progress of DNA replication in real time. It involves sort of labeling the DNA with 2 sequential halogenated nucleotides. So these are basically modified DNA nucleotides which get incorporated as normal, but which we can direct via fluorescent probes subsequently and then analyze by confocal microscopy. So if we just analyze how DNA replication forks progress normally in Mernib deficient cells, you can see a nice shift to the left here in terms of the average length of these bits of DNA. So it tells us that the replication is proceeding more, perhaps slowly in the absence of Mernib, but also perhaps that there are lots of stopping and starting happening. So we call that this form of replication stress. And it's consistent with Mernib having a role in DNA replication. Then we also moved on to check out an assay which we call the degradation assay, which is essentially involves this dual labeling for equal amounts of time, and then measuring and then we stall the fork using this drug hydroxyurea, which prevents further replication. And we can really then look over time to see how stable this bit of DNA is essentially. So if the DNA is stable, as you find in normal wild type cells, then these 2 labels are the same length. If it gets shorter, as you can see here in Mernib deficient cells, it means that MRE11 or some other enzyme has started to chew up this piece of DNA. And we can see this is exactly what's happening in Mernib deficient cells, because here in our Mernib knockout, we have a reduced ratio and this is restored by a chemical inhibitor of the MRA11 nuclease. And we see this in two different genetic backgrounds, and also by siRNA. And furthermore, so we mentioned that this happens at sites of remodeled DNA replication. And just to prove this is the case, we can actually reverse this degradation phenotype by depleting an enzyme called SMARCAL1, which is a DNA translocase that actually flips the DNA into that chicken foot structure. So this result essentially tells us that DNA needs to be flipped into that structure before the digestion that happens in MRNP deficient cells can occur. And similarly, whenever we deplete this factor SMARCAL1, the levels of DNA damage in MRNP deficient cells starts to go down. And we start to look at the how MRNP and MRA11 interact, and most of our work really suggests that we don't have a good handle on this yet, and this is something that we want to figure out over the next coming years, because we can see if we delete multiple parts of MRNP and try to look at the interaction between MRA11 and MRNP, that actually there is still some left no matter which bit of the protein we chop off. So it's something we really want to get a handle on over the next while. But kind of key to where we're at the minute is this assay, because we were really keen to try and see whether or not MRNP could actually physically impair the progressive the ability of MRA11 to chew up these bits of DNA. And so we've done this in vitro with essentially just a labeled piece of DNA and some recombinant MRNP and MRA11. So we can see here in untreated samples we have unmolested DNA that's completely one single band and not digested into smaller fragments at all. When we add MRA11, you can see this mostly gets chopped up into smaller and smaller pieces of various sizes. But when we start to add MRNP back in at greater and greater levels, we see that this degradation starts to occur less and less. So it does really suggest that MRNP can interfere with the ability of MRNP to digest DNA. And this is interesting because it's completely the opposite phenotype than we find at ionizing radiation and just double strand breaks. So there's something that we don't understand about the context of replication sites and double strand breaks that makes MRNP act rather differently at both sites. And so finally, I'd like to go back to what we sort of said near the start whenever we were looking at those BRCA1 and 2 mutant cells. So as we said, patients with BRCA1 and 2 mutations are really responsive to PARP inhibitors, and that's presumed to be due to the role of BRCA1 and 2 in repairing DNA breaks. But now there are these other functions coming out, the function that we just described, at reverse replication forks where BRCA1 and 2 is protecting that new DNA end from MRA11 mediated degradation. But also in more, in the past few months even, there have been work from ours, which is unpublished at the minute, but also work from several other labs, including Alberto Ciccia's lab, Sharon Cantor and Alessandro Vendini's lab all in the States, who has suggested there are actually more functions of BRCA1 and 2 in the context of DNA replication, and that these may well be associated with PARP inhibitor sensitivity. So we have the description of a new phenotype, essentially, where BRCA1 and 2 are considered suppressors of DNA gap formation during DNA replication. So if we go back to that diagram of DNA replication that we looked at before, we saw that whenever the cell hit the stop sign, it hit the brakes, or when the fork hits the stop sign, it hits the brakes and reverses and forms a chicken foot structure. Well, that isn't the only option open to it. So another option open to it is a phenomenon known as repriming. So the cell can basically say, okay, there's a block here, but we need to get replication done. So let's hop over it and start on the other side. And so this seems to happen more than we previously appreciated, especially in certain types of cancer. And so the enzyme that mediates this is an enzyme called PrimPol, which stands for primease polymerase. And that's because it can reprime on the other side of the blocking lesion and and restart polymerization of DNA on the other side again. And quite a few labs are now hypothesizing that this is an important contributor to sensitivity of BRCA1 and 2 deficient cells, because a number of observations suggest that BRCA1 and BRCA2 suppress the formation of these single-stranded DNA gaps, but also that PARP1 also functions to suppress those gaps for that gap formation. And some folks have hypothesized that these two independent kind of units of gap suppression are tolerable. Loss of any one of these two units is tolerable by the cell individually, but together the level of gaps becomes toxic, and this is hypothesized to be a real contributor to PARP inhibitor sensitivity in patients in ovarian, breast, and pancreatic cancer patients with BRCA1 and BRCA2 mutations. So in the context of MERNIP, this is really relevant as well, because we actually we conducted a number of experiments to show that MERNIP cells are actually sensitive to PARP inhibitors like olaparib-2. And you can see this here in the sensitivity profile, but also on the right-hand side measured as a marker of DNA damage. So we have no real DNA damage in normal wild-type cells treated with olaparib, but a much higher increase in the level of DNA damage in MERNIP deficient cells. And you can see this sort of exemplified by a cell with DNA damage here, where every one of these little dots is essentially a site of DNA damage. We conducted essentially a modified DNA fiber assay to try and start to look at the formation of all these different gaps, because we figured that the gaps are extended by something, and most of the previous work suggests in BRCA1 and 2 deficient cells, it's actually MRE11 that's digesting these gaps. So again, this nucleus is coming in, and where these gaps form, the MRE11 is acting on them, perhaps completely aberrantly and in a rather out of control way. And so we know, we've long suspected MERNIP of being something that controls MRE11 activity. So it makes sense to start to look at these gaps in the context of our MERNIP deficient cell lines. And again, we've used our MERNIP knockout CRISPR cell lines and modified a DNA assay so that we add the PARP inhibitor first, then one DNA label. Then we add cisplatin to introduce some DNA blocks. And then following our second label, we add this nuclease called S1, which is only able to digest single-stranded DNA. So, and then we measure the length of this DNA tract. And so when the tract gets shorter in the presence of S1 nuclease, it suggests that there is single-stranded DNA there, which suggests that a repriming event has occurred and a gap formation has occurred. And if we look at the right-hand three data points here, we can see in a normal cell, we've got X amount of tract length here. This is much shorter in both BRCA2 and MERNIP deficient cells treated with S1 nuclease, so suggesting that there are extensive gap formation in the absence of either one of these two protective factors. And if we go in closer and start to analyze the role of MRE11 in the process, we can see here, if you focus in on the right-hand side here, we have our MERNIP knockout cell sample, and that's getting rescued whenever we add a chemical inhibitor of MRE11. Similarly, if we have our BRCA2 knockout sample, that's also getting rescued when we add our MRE11 inhibitor. So it looks like in this case, there's a sort of phenotypic copying of BRCA phenotypes by MERNIP deficient cells. So it really looks like MERNIP is controlling MRE11 perhaps at these gap sites to prevent their extension and to perhaps prevent their closure. And again, we mentioned that the enzyme that does the gap formation is called this Primpol enzyme. And if we concentrate on the right hand side, we can see shorter DNA tracts in the absence of MERNIP after the S1 treatment. But then whenever we deplete the cell of this enzyme Primpol, we get really significant rescue in the length of these DNA fibers. So it suggests that it is this kind of repriming event on the far side of DNA lesions that's driving the single-stranded DNA gaps that are formed in MERNP deficient cells. So our conclusions are really just that we already had some really nice idea over the past few years that MERNP is really important for survival of cancer cells during replication stress. And this goes for classical replication stresses like this hydroxyurea drug that we looked at, as well as for other more cancer relevant chemotherapies that interfere with DNA replication, like platinum agents, PARP inhibitors, and also other replication stress agents. So I think we are sort of edging closer to figuring out exactly how MERNP regulates MRE11 at these sites, but it's becoming clear that both in the mechanisms of protecting these reverse fork structures and also in the protection of these of single-stranded DNA gaps, the MERNP is another crucial factor. And we want to start to delve into this a little bit more mechanistically over the next coming year or two, where we're going to really try and see if we can sensitize MERNP deficient cells further by inhibiting the error-prone polymerases, and these are the enzymes that fill in these gaps. So if you essentially make the situation worse, do our cells get more sensitive? We also want to try and address where MERNP is preventing these gaps from forming. Is it something that happens behind the replication fork or later in the cell cycle when bits of DNA can get that have not been replicated properly can get filled in? But also to ask the question, where these gaps actually come from in the first place. There must be a DNA end presented there for MRE11 to act on, and this may be something like a basic fission repair intermediate. So we really have this kind of triple function over the next few years that we've been funded today, and you managed to make some progress in, is that really just addressing how does MERNP function at all these different sites, and then we've also just begun attempts to do some structural work where we actually map how MRE11 interacts with MERNP. And that may be mechanistically insightful, but it also might create a potentially targetable interface. So classically inhibiting enzymes like MRE11 are it's quite dangerous because it's an essential enzyme for life. So it's a difficult target to drug in cancer cells. However, if we can inhibit the interface between something that negatively regulates MRE11, then potentially we can we could exacerbate gap formation in cancer cells. For example, in very specific scenarios that might allow us to improve the response to PARP inhibitors and other replication blocking agents. But what we're also really getting into now is it's just looking for the first time and trying to identify MERNP deficient cancers. So every cell line we looked at for years, at least in the cell line level, had got plenty of MERNP, and it was rather unusual because you normally see a little bit of a spread in terms of the expression of many proteins in cancer, just by the nature of the disease. But we finally realized that there is actually a subset of ovarian cancers where MERNP appears to be underexpressed, and I think we've analyzed around 15 cancer cell lines so far and found that in 2 out of the 3 high-grade serous ovarian adenocarcinoma cell lines that there's no detectable MERNP. So there could be a subset of MERNP deficient ovarian cancers out there that might be predicted to respond well to therapy, but which may have on the basis that they don't have MERNP, but they may also have they may, to all intents and purposes from a genetic perspective, appear not good candidates because they have no mutation in BRCA1 or BRCA2. Okay, so I'd just like to finish up there, and I just acknowledge everybody in the lab and our collaborators in Switzerland, Oxford in the States, and also UK, RI, Northwest Cancer Research Wales for funding us. So thank you. I think you're on mute, Mairead.
I'm on mute, sorry. I had unclicked myself, but it hadn't worked. So great talk, Chris. Thank you very much, very much. I particularly loved the photo of you as a proud CRUK postdoc when you kind of identified mRNP as the novel DNA repair.
The stress, the stress wasn't apparent in photos.
You looked very proud. Right, we've got some questions. So from Kate Lydiard, there's a couple of questions. So we'll start with what regulates mRNP expression, and are there any mutations or epigenetic modifications in the human population or in the cancer patient population? So I think that's a really good question.
It is actually, and it's something that I've literally just thought we've really ignored this because we kept looking in cancer cell lines and finding absolutely no difference in their expression, but there are clearly some out there. So and we have found them quite recently that there are some lines that with impaired expression, and we want to really do this as a precursor to IHC. And maybe looking at tissue microarrays, but we need a nice antibody to do that. But yeah, I've just told the postdoc to go and delve around and see if it's the promoters methylated in some ovarian cancer samples, because there's so much information online now, isn't there, informatic databases. So answer to your question, yeah, there could well be some methylation or something like that. But again, we don't know. We don't know why this has gone in these lines yet. But yeah, there are multiple mechanisms, but methylation, yeah, or something like that surely could definitely be one of them.
So it could be epigenetic modifications, or it could be deletions.
Yeah,
but mutations in the cells. You don't know.
Yeah, I don't think it is. I don't think it's a deletion.
Okay.
Just looking at the karyotypes of all the lines that we have. But yeah, but again, it's a bit of a mystery for us at the minute. So, but we don't want to. Yeah, you might think methylation or something is probably pretty likely.
All right. Okay, so watch this space. And the second question from Kate, do you see the same elevation of DNA damage in a double mRNP and PARP inhibitor knockout, or is the effect of the PARP inhibitor distinct?
It depends on which drug, I guess, is the question. To which drug is the for DNA damage? It's a good question. We've never checked, but I guess it depends also if you're looking at background damage or whether you're looking at damage in response to a particular agent. If it's something like PARP inhibition, then the PARP trapping functions of PARP inhibitors could sometimes be ablated by doing it in a PARP1 knockout background. I think maybe that's what Kate might be sort of alluding to there. But yeah, we haven't checked basically. So.
Okay, Kate, if you've got any follow-up questions, follow up in the chat. So Duncan has just said, great talk. And then Owen Gallimore has a question. You mentioned targeting mRNP. Is this something you'd anticipate working best with a PARP inhibitor, or in the case of PARP inhibitor resistance?
Well, I mean, I think it's tough to say because I don't think, although we have evidence that the AHR homologous recombination doesn't work quite as well in MERNT deficient cells. The knockout mouse is actually alive, so things that are absolutely essential for homologous recombination don't generally survive past the embryo stage. So I think these mice are infertile, which is suggestive that there is potentially a lot of DNA repair or DNA breaks going on in the testis during sperm development. So it does suggest a DNA repair phenotype, but again, it's not quite. I don't think it's quite on the level of BRCA1 or BRCA2 knockout in terms of sensitivity. So, I don't like to predict what will happen with the cancer as well either, really, because at the end of the day, these are all cell line models. But I think the things that we see it most sensitive to seem to be replication blocking agents of multiple types, not just PARP inhibitors.
Okay, great, thank you. Okay, another question. So beautiful work from our next question. Two questions. Does mRNP regulate both endo and exonuclease activity of MRE11? So that's the first one.
No, great. Well, these are all great questions, actually. It's a good question, and the answer is we did do the blocked end. There's a block. If you block the end of the DNA substrates that we use for nuclease assays, then it essentially forces the MRE11 to do what it does in vivo, which is do the endonuclease cut first. And we have done those assays and we don't see any difference. That would, I mean, we know that MRE, it doesn't make it's not a perfect representation because in a tube the MRE11 will cut anyway. Even if there is something there that will regulate it or not, and it's not a perfect recapitulation of what happens in vivo at all. But I guess on the most superficial level, the end degradation that occurs at reversed forks is mostly considered to be exonuclease cutting. So I think I don't want to speculate too much about the endo function, but I think the exo function is it's definitely a good candidate for being inhibited by MRE11.
Okay, and then the second part of the question was RNF is an iron ? Binding protein. Is mRNP iron binding constitutive?
Yeah, I think I'd love to know that actually. But
We'll have to come back next year and tell us the answers to all these questions.
Well, we're a little bit confused at the minute, to be honest, because I skipped past this result just because it was running that short time, but we had a quadruple mutant of the cis4 cysteine residues that are predicted to coordinate the N-terminus, or predicted to in the N-terminus to coordinate a metal ion, and that mutant functions just as well as the wild-type protein in protecting replication forks. So either that, but I guess there is a possibility also that sometimes you can have histidines involved in zinc finger motifs and things like that. So it's possible that we've got the wrong ones, but or it's possible that just in the assay we've used isn't important. But yeah, I think again, if we get any kind of structural data on this that actually does suggest it binds an ion, then it would be it's going to be really helpful in actually figuring out what this thing does. So.
Okay, and then a couple more questions from Kate. mRNP is very close to 5q telomere, ? Do the ovarian cancer patients with reduced mRNP expression also have mutated H-TERT at 5p?
Oh, I don't know. So I will definitely check now. So,
there we go.
Yeah, it's interesting that it's very close. I mean, I looked around for alterations where that cytogenetic band, so it's 5q35.3 is affected, and I didn't really. There aren't too many syndromes out there that are associated with it or anything like that. But I would say it's actually very close to RAD50 as well, which is one of the members of the complex. So it depends how much is missing. It depends how much is missing, but there's if there was a big chunk of DNA missing there, I expect that would based on the other genes that are around it, if there was a whole section of the DNA gone, then it would probably invalidate cell viability. So.
Okay, and then one question, then I have a more clinical question. Is there a microRNA that might regulate mRNP?
Oh yeah, I'm sure there is. Yeah, it's good, but I mean, I've never gone into the world of microRNA, but I fully.
They're topical at the minute.
I would say I've had a quick look a while ago, a couple of years back, on some of the software that predicts whether a microRNA will target a certain factor, and the list was fairly extensive. I think there were quite a few that in theory could target it. So yeah, I think it's something I think we've never gone near and probably should do in the future, because it's a level of regulation that could be very important.
Okay, and then just one final question. Then just wondered if chromosome 5 destabilized particularly in these mRNP ovarian cancers.
Oh yeah, I don't think that. I mean, I think I'd have to look in real detail at the whole chromosome. I just in terms of where I looked, I essentially looked at is that part of the chromosome still there? And it was. So I don't know if I can't really sort of speak more widely about the integrity of the chromosome in general, but yeah, my main interest sort of ended when I saw that it wasn't missing. So no, that's right.
So Kate has thanked you for answering all her questions. I just had a slightly more clinical question. So, you know, along the way, how would you potentially plan to develop a treatment to selectively target, mRNP in cancer cells, but not in normal cells? That would be the ideal scenario, right? And what kind of would be the timelines of potentially doing that?
Well, I guess the couple of angles on it essentially are, it could be, I don't really like the term, but it could be a bit of a BRCA factor. It makes it, that there, firstly, it could be of more immediate relevance, so that there might be some patients out there that would respond well to certain drugs based on their MRNP status. And that would be so it would inform sort of practice a little bit more potentially. So, but a lot of work needs to be done on that. We need to, this is very early days for us. To answer your second question, I think this is why we're really trying the structural stuff. And to be honest, it's quite a risky strategy because this is a very difficult, complex to visualize. We're trying to do it with the human version of mRNA complex, and it's notoriously finicky to get structural data on, although we are going to collaborate with some really great folks who are fantastic at it. But yeah, if we can map that kind of interface between the two. So it's kind of like I said at the end of the talk that mRNA-11 inhibitors are a fantastic target, but there's got to be a lot of off-target toxicity there because it's essential for DNA repair and for replication fork restart if a fork stalls and so forth. So you're where I think the beauty of targeting factors that can control mRNA-11, it comes in is that you're not actually targeting mRNA-11 itself, but you are targeting the processes that and the control of mRNA-11. So I think that's a big advantage, and actually mRNA seems to be most well expressed during embryogenesis, and then it actually shuts down in quite a few tissues. It's not gone, but the expression levels do drop in adult tissues and really restricted to the brain and testis. So if you get a drug that doesn't cross the blood-brain barrier, then your main worry is probably fertility, like the mouse knockout has got a fertility problem, for example. So you kind of take it, hopefully just through having less in other tissues, you can take some of the, maybe some of the off-target effects off. But I think we're really hoping to target, if we could figure out where mRNA-11 and MRNP bind, it's a bit of a finger crossing moment, but you think, well, maybe it's a targetable interface with a little pocket or something like that. Although we have made some nice advances in recent times in targeting more sort of flat interaction surfaces too.
Okay, thank you very much. So it's just 1 minute past 4, so I'll close the session. I'd like to thank Owen Gallimore for organising, doing all the hard work, for giving me the opportunity to chair this afternoon, and Chris the opportunity to speak, and thank Chris, of course, for a fantastic talk and for answering all our questions. And then to thank you all for attending. It's half term, so we've had a fantastic turnout considering that. So thank you very much.
Okay, thanks everybody for listening.
That was really nice, Chris.
Oh, thanks. Are we in a